Welcome to the Drug Discovery Initiative DELopen in vitro award funding initiative. My name is Salvatore La Rosa. I'm the Chief Scientific Officer of the Children's Tumor Foundation. And today we have the pleasure to have as a co-presenter Letian Kuai, Senior Director, Head of Biology and Informatics at WuXi AppTec. Together, myself and Letian will give a presentation about the awards itself, how you're going to use the funding and the technology and information about DELopen. We hope that at the end of the webinar, you'll have all of the answers you need to submit an application to obtain this award and use this technology for neurofibromatosis research. So let me move the slides and show you what the webinar outline is. I will give you a brief introduction of the CTF DDI award program. I will also tell you how this program came together as a partnership between CTF and WuXi AppTec. I'll talk about the scope and the award details, what is the workflow, the use of funds, and some notes and award dates that are always useful. Then I will handle it to Letian. It will give a presentation on the DELopen program, what can be done with this technology, some example of key studies, and so on to give you some ideas. And then at the end, we'll have a Q&A led by Rebecca and Vanessa from our office. So you're very welcome to type at any time questions in the chat. So there is a button at the end where you can see there is a chat, and they will lead that, and we'll promptly ask the questions and we'll answer. Okay, so as a brief intro, the first CTF DDI program was launched in 2006. This has been always our translational, let's say, award type, where we provide some funding for research that needs to address very important questions of screening drugs or a finding model that needs for a translational research. Since 2006, we've been awarding 76 grants for a total of 2.86 million. This award program has been very successful. Awardees have received follow-on funding for more than 5 million from other agencies, including NIH, CDMRP, and CTF as a result. The application turnout time is very short. Normally, this program in the different definitions ask for a 3 to 5 max page of proposal application. And the review time and assignment is always kept to the minimum so that we can provide funds to researchers very quickly. Through the years, we've been having different variations of the DDI. We started with in vitro awards, but now we expanded to also in vivo. And we launched a version which is the register reports that we just concluded at the beginning of this month. And we also use this program for the NF2 Accelerator awards that we launch again probably throughout 2020. So following the partnership between CTF and WuXi, this specific award program started as a collaboration and an idea to leverage on the DELopen program that WuXi has launched and allow our research community to get some funding to run this program efficiently. So the key point here is that for the first time, we are opening this this award in conjunction with an external program that will allow new technology, hopefully, to be applied to the NF field. So is the eligibility criteria has to be that it's not only experts in the NF fields can apply, but also any researchers that has a valid hypothesis and capability that is working on a target that is relevant for NF can apply for this fund. All of the award page details are listed at the bottom or at the end of my bullet points there. So if you go to ctf.org and check on the research page, then you'll find the Drug Discovery Initiative DELopen Awards with all of the details. I'll come back to that later. So what is the scope of the award? So the focus of the request for application is to support exploratory and developmental research projects, including the identification of new chemical hits for specific targets involved with key aspects of NF biology. And here we listed 3 examples, but they are not limited to this. Of course, the focus of the award is also linked very closely to the technology of the DNA encoded library. So technology can be used to identify early hits, for instance, for novel targets, or we can, you can think about identifying tool compounds for targets that have no known small molecule and create those tools for the community. Or even for existing targets, you may think that the existing inhibitors that are already being identified, they are not ideal, and you want to identify new chemical class inhibitors for those targets. The funding scheme is our DDI in vitro, and applicants can request up to $40,000 for the duration of the in vitro awards that will be 12 months. Of course, as all of the CTF awards, the policy that we allow max 10% indirect cost to be included into the maximum of $40,000. So how is the workflow and how can you use the funds and what are the main outputs? On this scheme, I actually depicted what is, how this award will work. And if you see in white are basically some process that are provided by WuXi at no cost, and in green highlighted those processes there where the funding can be used. So everything starts then when we select the applicants and we provide the funds, they can apply to WuXi to get this kit of DNA encoded library. Then the kit comes with a specific instruction on how to run the affinity screening or how to run any type of screening. And even if the instructions are, well written, you always have the opportunity to contact directly the scientist and Letian or someone will be in charge for that, where you run the screening. And then after the screening is run, the research returns the selection samples to WuXi, and WuXi will analyze and decode what are the compounds that actually were binding to the target. So WuXi will summarize the affinity results, and the affinity results will be, of course, shared with the awardee. And depending on, there will be positive or negative results. The idea will be either, to stop, terminate, or thinking about, a different approach. Or if we have positive results, then the next step will be the resynthesis of those positives. And the DELopen program offer up to 5 compounds to be resynthesized at the price of $2,000 per compound. The compound will be synthesized in 10 milligrams quantity, and it will be sent back to the awardee for biological validation. And of course, biological validation will be in the same affinity screening, but also you can think about orthogonal screenings or functional screenings. At the end, if the biological validation will not validate those hits, of course, the project will terminate, will be unsuccessful. But if some hits are validated as inhibitors or binders, then we'll come to the phase of the chemical decoding agreement, where the structure will be revealed, and there will be some options for how to progress the research. Of course, what is really important and what I think for this type of proposal has to be the focus is the affinity screening. As you will see, and use of funds, that is on my next slide, this is the core of the proposal. You need to think about how to create an affinity screening where you can use this DELopen library. Then, of course, the affinity screening is also important to validate the resynthesized compounds. So this is, I think, where focus has to be, sent. Funds can be used to, of course, cover all the costs involved with the affinity screening. And if you think about maybe a novel protein or a novel that you need to synthesize or produce from scratch, we also allow working with a CRO that is specialized in protein production. So you can partner with anyone that will allow you to generate those reagents that are needed for the affinity screening. Then, the use of funds can be for covering the cost of hit resynthesis up to $10,000. Actually, the DELopen will allow you to resynthesize 5 compounds, so that's the $10,000. And of course, the cost of biological validation, that is testing of newly synthesized compounds with orthogonal screening as well. What the funds cannot be used for is to negotiate the chemical decoding agreement. You'll see that there is an option where you can actually buy out the results altogether if there is some interest in patenting those compounds for further development. And you cannot use those funds for targets that have no clear connection with NF biology. So anyone can use this technology and can access this grant, but has to be a target with a clear connection with NF biology. Some important notes: WuXi will not know the identity of the target, so they will not have any participation in the review of the proposal, and the decoding of the beads that will be sent back will be done in a blinded fashion. Applicants also will not know the identity of the compounds until after the chemical decoding agreement will be in place. So there will be a report of activities on, let's say, compound number 1, 2, 3, and 4 with all of the other data. But this part will be, so the decoding of the structure will be done after the chemical decoding agreement. And what is really important is this: independent from any option that you will go for on the chemical decoding agreement, WuXi will not claim or require any IP on the newly discovered compounds. So here I will leave to Letian maybe to explain a little bit more, or we can answer specific questions. But the idea will be to encourage data sharing. So WuXi will allow for the 5 compounds to be released as the structure revealed to the applicants, if the applicants agrees to share the results of the remaining of the screening. Of course, those 5 compounds will be taken out of the library. Or, as I said before, the option to obtain full data and exclusivity for a fee, and there is a buyout option that can be also considered. So proposal will be selected and prioritized based on feasibility of the affinity screening. And of course, the relevance of the target that is proposed in the proposal. In terms of award dates, the form is going to be available through our normal proposal central hub, clicking on grant opportunities, filtering by Children's Tumor Foundation, or typing in the foundation name in the search box. Applicants will then, click on apply now and start the application. And the application will be online from January 20, which is next week, and will stay on until March 9, which is the day by which we'll close our acceptance of the proposal. It will start, the review process. Again, at the bottom is the URL for all of the informations about, the guidelines. All of the information that I presented here can be found there. So now it's my pleasure to hand it over to Letian, where he will present the technology, and will please write your questions again in the chat. We'll answer at the end, or if there is any burning question, we can answer while we go. I'll stop here. Rebecca, is there any questions so far on my part?
Yes, there's a question. Salvo, can you hear me?
Yes, go ahead.
Okay, there's a question from Mr. Sarnoff. Are the exclusivity agreement terms defined up front? Is the buyout option cost something you can share with us?
Yes, this is, if you go to the DELopen, information is publicly available. I think the fee for a complete, buyout of the results is $50,000. Of course, the option, instead of sharing the remaining of the results, is, it's, of course, it's free. There is no cost on that part. And of course, the 5 structure that will be resynthesized, those will be kept confidential and will not be communicated to anyone else. So those 5 structure will be taken out from the library, and those will not be screened anymore, or will be taken out anyway and not included in future results. Letian, you can confirm on this part. Of course, this is more like on WuXi program, on the DELopen program, but I think it's very clear. So there is a data sharing opportunity at no cost, where you share the remaining of the results, but not the 5 top results that's, is going to be resynthesized, or you can decide to buy out the whole screen, and then, all of the selected structures will be taken out.
Yeah.
Is that correct?
Yeah, I confirm this is absolutely correct. I just wanted to clarify that for the data sharing, and you're not sharing the data with WuXi, and instead that data will be accessible to a broader research community. So everybody can potentially have access to the database. There will be some controls. For example, the future accessor, they will not automatically have the right to see the structure without the original data generator's consent. Okay, what they can see is the selection profile, for their data mining needs. So the credit of these data still belongs to the original data generator, and they will be part of the future collaboration if anything interesting should come out of the remaining data.
Okay, any other question?
No, not at this time.
Okay, so I will stop sharing, and Letian, you can start your presentation.
Okay. All right. Let's go to So, to go to the front page. Can everybody see my screen?
Yes, we see your screen.
Okay, so my name is Letian Kuai. I'm leading the DNA encoded library selection platform and informatics platform at WuXi AppTec. And today I wanted to give you a brief introduction of the DELopen program. And I will start with some general introduction to the DEL technology. And then a little bit about the core engine of the DEL platform at WuXi. And I will explain why we wanted to generate this DELopen platform. And at the end, I will give you a case study with the DELopen libraries. As you all know, we as human beings, we have been searching for medicines for thousands of years. And we have made very interesting cycles. At the beginning, we looked at herbal medicine and screened different herbs, and noticed that some of them can be used as medicine. And then we thought we have mastered that technique, and then we invented the alchemy, and then we started designing drugs or a substance that can prolong life. Some notable examples in there. And then, and lately we realized that we still don't know enough about the biology and chemistry. And then that actually gave birth to the high throughput screening on chemical compounds. I'm one of the those who believe that ultimately we will arrive at a stage that we can rationally design medicine with artificial intelligence. But between now and then, most of the medicines are still, it's fair to say, most of the medicines are still discovered by accident. Okay, so I think one important question is, for the time being, how do we make these accidents to happen faster or surely to happen? So here is a DNA encoded library which tries to tackle this problem from a different angle than the high throughput screening. As you probably already read from the literature, the DNA encoded library is based on a combinatorial chemistry approach where the compound can be synthesized in a split and pool technique, so that you can arrive at millions or billions of compounds in a simple synthetic scheme. The difference in the DEL encoded library, the DNA encoded library, is for each of the synthetic step, for each of the building block, we assign a unique DNA tag to be associated with that compound. So at the end of the synthesis, we have a library that has billions of compounds, and each of the compound is specifically barcoded with a DNA sequence. That gives us a unique way to deconvolute this library after selection. In contrast to the traditional way of using mass spec, we can use the next-gen sequencing to read out the DNA barcode and to figure out which compound is binding to your target of interest. Okay, and that also gives us a very, very simple affinity selection that everybody can run on their benchtop. It does not require heavy instrumentation because all the small molecule the library molecules are in one mixture. So basically, you only need one tube to conduct this selection process or screening. Okay, and it has a very fast turnover time. It should the selection process takes about 1 to 2 days to complete, and it does not require lengthy assay development or validation, as long as you have a protein that you can confirm the folding and activity, and it has a affinity tag, and you're basically good to go. So this technology has quietly evolved for decades, and so starting from 2000, there are companies working on this technique. And obviously, the original idea was in 1992 when Dr. Rich Lerner from Scripps initiated this concept and demonstrated that this actually can be done. It was not until 2000 when when different companies started taking this idea and turn this into a practical DNA encoded library. This obviously requires a lot of upfront infrastructure investment, including the building block collection and the automated synthesis, and then the data decoding. And obviously the next gen sequencing was the last push for this technology to bloom. Okay, and so the next gen sequencing can basically survey millions to billions of sequences in one sequencing effort. Okay, on this slide, you will see various of structures reported by different industry or academic labs of successfully identifying nanomolar hits for targets from different classes. And these compounds, they're at different stages. Some are at early stage discovery, and some have gone into the clinical trials. Okay, and I also wanted to mention with DNA encoded library, not only can you use it to find a drug candidate, and if you think about the anatomy of this molecule, you're actually screening small molecule in already conjugated format. So, and then at the end of the day, you will identify a small molecule ligand that has affinity to your target. Therefore, anything that's associated with affinity, you can also use this technology to discover. This will include protein target purification or biomarker identification, or you can even think about the drug delivery, and where you can find affinity ligand that will bring your drug to anywhere that you want, or to extend the half-life of the existing drug. It can also be used as an imaging reagent for you to enhance the histochemistry, and also may be helpful in diagnosis. To the right of this slide, I'm giving you a very, very simple example where we can identify a small molecule that can drag a piece of DNA inside the cell. Okay, and so this was identified several years ago at GSK, where this small molecule can successfully bring in a piece of DNA that is about 100 base pair long inside the cell. Okay. So I want to switch gear a little bit to the WuXi DEL platform. This is basically the engine of the DELopen platform we're going to talk about. At WuXi, we started building the platform in 2017, where we focused in making the DNA encoded library. And to date, we have collected about 240 libraries of various chemistry and various collection of building blocks. In 2018, we officially launched our selection campaign business. And to date, we have 120 scientists And in 2019, we have served more than 40 business clients, and we conducted selection campaigns on more than 80 projects. The service we provide actually also include the library production service, where a client can commission us to make a specialized library. Although that is not in the scope of a DELopen, but anyone who are interested in this can feel free to think about how in the future, how do we customize the required service. To date, the main platform, like I said, we have seen a total of 82 target, and they expand across 12 target class depending on how do you cluster them. And I'm proud to say that we have seen the success, quite remarkable success rate in different target classes. In the main bar graph, what's shown here are the percentage of a target that will yield a informatics hit. And from the informatics hit, and we use the binding confirmation as our secondary assay, and we have about 70% of confirmation rate. And on average, our success rate is about 85%. Okay, so this is the core engine that supports the DELopen platform. So now, so what inspired us to design this DELopen platform? It was inspired by a perspective article written by Professor Richard Lerner, and he asked this question, that DNA encoded library, when it's first designed, it was very suitable for any researcher to conduct a screen at their bench top. But why in decades people have difficulties accessing this technology? And he also asked this important question of how do we form a new relationship between industry and academia to let this technology to be accessible to academic researcher. There are a few pain points in this technology. For example, as I mentioned a few minutes ago, that the synthesis of the library itself is a huge undertaking. Yes, the academic labs usually does not have the resource to acquire thousands to tens of thousands of building blocks to make these libraries. The cost of the library construction is very high. And also the infrastructure to allow the storage and maintenance, and also the data mining when it comes to the selection result. The selection result generated by next-gen sequencing is also a huge task. Okay, so for us to solve this problem, we took a hard look at the DEL, the DNA encoded library workflow, and we realized that the synthesis of the DEL library and also the data interpretation and the storage, these two big ticket item can be centralized, and what can be distributed is actually the affinity selection and the resynthesis and the confirmation. So we have decided to change the format of the DEL, and then we will synthesize the library and then send the library aliquots to academic users as a simple reagent, and then the researchers can use the reagent to conduct their own selection. And this actually solves several problems when it comes to target exclusivity, and also the structure revealing, because now the researcher has the opportunity of conducting the selection, but without revealing the target identification. Okay, so we see that as a huge advantage for this technology to be broadly accessed by the academic researcher. So here, and you see this slide page that describes what's typically involved in a DEL screen. When you have a protein, you will have some method to validate that protein is correctly folded and has a desired function, and then you can immobilize that that protein onto a affinity metric, and then you incubate the you incubate the protein with the library, and then you wash this complex, and then you elute the binders, and then you repeat this process several times, and you will have the selection outcome, and then this is ready to be sent back to us to do the sequencing. And we will report back the data analysis. So we will tell you if this selection has been successful, meaning do we see significant outliers in the library or do we not? If there is no significant outlier, we say this is a unsuccessful selection. If there are outliers, we can proceed to the next step of first revealing the selection profile, and then we can discuss the method of confirming some of the hits. You can choose to confirm either off DNA confirmation, as Salvo mentioned, for $2,000 per compound. You can also choose to do the on DNA confirmation, where we can quickly make even smaller amount of the compound on DNA to confirm the affinity. Okay, and in the last part I wanted to describe to you is a case study. We've already test drove this platform with a few collaborators, but for privacy concern, we cannot tell their story. And here we chose one case that is during our internal testing of the DELopen library. We conducted some selections against a panel of target. So here is a target protein. It has a molecular weight of 69 kD, and we cannot describe what target it is, but it is a his tag, it is a full length, the origin is human. The selection we did here is fairly simple. We have the protein, and we have a no target control, which is essentially the affinity beads itself. In the actual DELopen kits, you will receive 4 copies of the library, which will allow you to conduct 4 comparative conditions. But in here, there's only 2 conditions and repeated. Okay, as you can see, the Venn diagram, the selection logic is very clear. We're looking for things that show up in the target protein, but not show up in the no target control. Okay, and for every selection report, you will receive a page somewhat like this, and it will describe the molecular attrition among different selection rounds, and you will see the sample workup report to show that we have successfully amplified your selection sample, and we purified it, and then the next gen sequencing data is with normal specification. and that is typically an indication of a good selection. When we go into the data analysis, you will typically see a plot like this. In here, you have 4 selection conditions, and on the x-axis, and in the y-axis, there is an enrichment that is a arbitrary unit that tells you how many fold of improvement of a certain chemotype above the population average. So and you can see on this line plot there are multiple lines, and if you see a line across different conditions, that means the chemotype has shown up in 1 or 2 or up to 4 conditions. And obviously in this campaign, the condition 1 and 2 are the repeated target selection, and condition 3 and 4 are the repeated no target control. So obviously you are looking for species that show up in condition 1 and 2, but not in 3 and 4. So after data analysis, we have noticed some SAR information, and we noticed the preferred structure is in cycle 2, which has a structure shown in purple here. The index number of building block 2 is 351. It also has a preferred structure in cycle 3. And you can see the index number of these building blocks. Okay, so with that information, we could informatically reconstruct the potential look of the molecule, and in here the cycle 1 building block is not essential, but the cycle 2 and cycle 3 highlighted in purple and brown. Are the sort of a decorative part of the cycle 1. Okay, so as you can see from a simple selection, not only you have a hit candidate, you will also see the family information, and some of them have a direct linkage to the future SAR. Okay, so based on that information, we proposed there are 6 compounds that probably worth making, and in the report at this stage you probably will not see the structure. However, you will be provided with the library ID, building block ID, and also the molecular weight and C-log-P, and a panel of other PhysChem properties that will help you decide, what are the compounds that have more potential of becoming a drug. Okay, in a second library. So what you saw in the previous 2 slides is the hits that's coming out of 1 out of the 5 libraries. On this page, there is another group that's coming out of a different library in the collection. As you can see, the story is quite similar. We're looking for things that show up in condition 1 and 2, but not 3 and 4. And again, the SAR analysis revealed a family of compounds that are worth making. And the hit confirmation, because this is an internal project, we did not choose to use the off-DNA synthesis, and we used a more direct assay that is closer to the selection itself. This is what we refer to as an on-DNA confirmation, where we use the library chemistry with the DNA to make this the compound on DNA, and then we use affinity mass spec to evaluate whether these compound would bind to the original target. As you can see, we tested a total of 6 compounds, excluding the last one that is a comparator. So out of the 6 compounds, we successfully identified 4 of them as a significant binder. Interestingly, the 4th compound here, we also noticed that there are the binding actually attributed back to some of the byproduct during the chemistry synthesis. So this is actually a interesting phenomena that, as you probably know, for a combinatorial chemistry, and you always make the desired product, and then you also make byproduct, because some of the reactions will result in multiple products. So this actually illustrate that the DEL library, the actual size of the DEL library is actually bigger than what it looks, and sometimes you could even have surprise findings, and some of the byproduct have interesting properties, and some of them are partial products that is even smaller than the original, than the desired product. Okay, if they turn out to be a hit, that actually helps your SAR understanding, and also some of them are truncated. That gives you an even better starting point for your drug candidate. Okay, so I'm going to stop here just to and give you a final summary of today's presentation. I wanted to say that WuXi had always been dedicated to provide cutting edge tools and services to the research community. And the DEL, I hope you're convinced that the DEL, the DNA encoded library, is a lightweight screening tool that allow researchers to rapidly access billions of compounds. And DELopen, we designed a platform that is aiming at solving the decades-long industry academia collaboration problem, and provide a open and rapid DEL solution to the academic community. And I'm going to stop here and ready to take questions if there are any. Thank you.
So thank you, Letian. Really.
Excuse me, I think there are some echoing. Can you repeat that question?
Yeah, I was asking about the size of the library.
The size of the library. So in the first in the beta testing, we provided 5 libraries, and then the total the total diversity of the 5 libraries are 3 billion. But in our official release, now we're going to release 40 libraries with a total diversity of 4.4 billion.
Okay, thank you. So let's say if we will award some of this DDI-DEL, there will be the expanded library probably will be ready to be used by the applicants, right?
Right.
Okay.
Yeah, so these libraries are provided in a mixture. So you cannot separate the library. So when you run the selection, all 40 library goes in at the same time.
Okay, Rebecca, do we have any further questions?
No, at this time, there are no additional questions.
Okay, so what I want to say is this webinar is going to be, has been recorded, and we'll add the recording to the DDI-DEL page as a reference for you to go back and maybe review, or if you know of any collaborator that will be interested in this program, they can refer back to this presentation and look at the video. If there are no further questions, I would stop here. And thanks everybody who attended. Thank you, Letian, for your presentation. And we're looking forward to receiving your application for this, we think, very interesting award. Thank you so much.
Thank you.